Hello, I am Deepshikha. Let's start with a quick revision of DNA replication process. First of all, DNA replication is the first step of central dogma incident, and this is the DNA to DNA production. Specifically, this DNA replication occurs during the synthesis phase or S phase of interphase during the cell cycle or cell division. Let's start the main process of replication. First comes the Gyrase enzyme, which mainly unwinds the supercoiled structure of DNA. When it unwinds the structure, then comes the Helicase enzyme, and the main function of Helicase is break down the hydrogen bond between the alternative nitrogen bases, and after the breakdown it looks like this, like a fork, which is termed as the replication fork. Next come the DNA Primase enzyme, which mainly form the primer. Primer is the main base of replication process, which identify the sequence, and it binds with the parental DNA strand and guide the DNA polymerase enzyme to form the new nucleotide chain. The main function of DNA polymerase enzyme is extend the new nucleotide chain, and always it has a biggest characteristic is that it acts always from the 5' to 3' direction. Then come the process, and after the primer binding with the parental strand, DNA polymerase enzyme acts from 5' to 3' direction. So generally it acts against the 3' to 5' parental DNA strand, and this runs smoothly, and for that reason the newly formed strand is a continuous strand, so it is called the leading strand. But when the DNA polymerase will act against the 5' to 3' strand of parental duplex, then it cannot run smoothly, so it forms like this fragment, and these fragments are called the Okajaki fragment, and with each Okajaki fragment there will be a primer. This type of continuous strand is termed as the leading strand, and this type of discontinuous or fragmented strand, newly formed strand, is termed as the lagging strand. Now comes the nuclease enzyme. This restriction nuclease enzyme is termed as the molecular seizure. Its main function is to remove the primer. It cuts the primer portion and remove it. After that comes the DNA ligase enzyme, which is the molecular glue. It mainly attaches the fragmented DNA portions and form a complete newly strand DNA. So after that we can see that from one DNA there forms two copies of new DNA, dotted DNA, and when you see this, newly formed DNA contained 50% of parental strand and 50% is the new strand. So it can be said that in the newly formed DNA or dotted DNA there have conserved 50% of parental DNA. So this replication process is termed as the semi-conservative process. If you have any query, please let me know in the comment section. Thank you.